Hello, my name is Rohan Khanna and my topic is Transcriptomics analysis of type 2 diabetes gene expression. Diabetes is a disease defined by blood glucose level fluctuation. Fluctuation in blood glucose level might not result in major symptoms over a short period of time, but chronic conditions may lead to vascular complications, both micro and macro. Diabetes is a group of metabolic diseases characterized by hyperglycemia resulting from defects in insulin secretion, insulin action, or both. In the case of type 2 diabetes, it's both, and in the case of type 1 diabetes, it's insulin action. The chronic hyperglycemia of diabetes is associated with long term damage, dysfunction, and failure of different organs, especially eyes, kidneys, nerves, heart, and blood vessels. In severe cases, necrosis also occurs in fingers as well as sometimes limbs. No cure for diabetes currently exists, but the disease can go into remission. Remission means that the body does not show any signs of diabetes because the blood glucose level is regulated. Although the disease is technically still there, that is, the symptoms do not show, but the cause is there. Remission consists of A1C levels below 6% as a significant factor. A1C levels indicate a person's blood sugar level over 3 months. Type 2 diabetes and its diagnosis. So, an impaired insulin secretion is the characterization of type 2 diabetes. This impaired insulin secretion from pancreatic beta cells is coupled with resistance of insulin in tissues of skeletal muscles, adipose tissue, and liver. An elevated blood glucose level. Diabetes can be diagnosed based on a measure of fasting blood glucose and or long-term levels of blood glucose, HbA1c proportions. A more detailed method for measuring the level of glycemia is A2, A2R oral glucose tolerance test or OGTT, which also allows the detection of impaired fasting glycemia IFG and impaired glucose tolerance IGT. Diabetes, a global pandemic. Over the past few decades, the prevalence of diabetes has exponentially increased worldwide. The statistics of 2017 shows that 425 million people worldwide suffer from diabetic conditions from that number 77 million are Indians. This concludes that every sixth diabetic patient is an Indian. The number of patients suffering from diabetes is not regressing but rather progressing, according to the leading researchers and the International Diabetic Federation. The International Diabetes Federation has established that this number of 425 million can or may reach 629 million in the next 2 decades or around 2045. The prevalence of type 1 and type 2 diabetes cannot be studied or estimated separately, as most of the studies are done with type 1 and type 2 diabetes, both samples together. But one estimate shows that developed countries have 88 to 90% of all the cases of type 2 diabetes, while 7% to 13% is estimated to have type 1. There was also a small percentage of individuals bearing other types of diabetes such as GDM or gestational diabetic mellitus, which happens in pregnant women. The research question which I took was, I wanted to explore and study the biomarkers for diabetes type 2 through transcriptomic analysis and to look at the pathways affected in type 2 diabetes. The data set I chose was obese insulin resistant humans with compensatory hyperinsulinemia dissociate liposis from glycemia as possible adaptive response to fatness. Now, what does that mean is that the data set we are using, first of all, is in our study is available on GEO website under ascension number of GSE 141432. So, the data set is divided into two groups, which is lean and obese sample, and the obese sample itself is divided into two groups as well. The overall design of this study was to shed light on mechanisms driving elevated levels of FFA and glycerol in the affected or obese group. And for this study, genome-wide RNA sequence analysis was performed. The the tissue present was subcutaneous wide adipose tissue, and the sample was taken from lean individuals or healthy or normal individuals, obese insulin resistant individuals, as well as obese type 2 diabetes diagnosed subjects. The total number of lean individuals were 9, and the total number of diagnosed or obese individuals were 16. My total sample size was 25. The data set was an expression profiling experiment done by high throughput sequencing, and the link between obesity, 5 blood levels of free fatty acid, with type 2 diabetes is not completely understood, and they investigated glucose homeostasis lipolysis in obese patients recently diagnosed with type 2 diabetes. The obese patients resistant to insulin who were non-diabetic subjects and the lean individuals. Age, sex, body composition, and fasting insulin levels were taken as parameters. The result of this study showed that the obese insulin resistant dissociate lipolysis from glycemic control. Now, the methodology. Analysis of the data was done through T-Bioinfo server. The data was fetched from NCBI website or GEO website, dataset ascension number GSE 141432. The data we took had some unique features. It had the data was divided, as I said, data was divided into two and then further divided into the obese sample was further divided into two parts as well. Through this we can understand my previous statement that the studies done on studies done on diabetes and obesity always have type 1 and type 2 diabetes together, linked together or coupled. I couldn't find a data set purely of type 2 diabetes or type 1 diabetes. The basic path we took was to collect the data from RNA sequence done by other scientists to make sure it was usable in our study. Then we processed the data, normalized it, and used it for PC analysis and DEG analysis. The results were then used for GO analysis, and finally we had our results, which will be further discussed later. The data taken had combined identity after the PC analysis. PC1 plus PC2 was more than 60%. and it had the data divided through body type classification. The PCA, or the principal component analysis, DEG, differential express genes, and GO, gene ontology analysis, were then done to find out highly expressed genes and important pathways. PCA analysis pipeline. The data was in the form of a matrix file where the values needed to be normalized. Quantile normalization was done using the TBioInfo server pipeline. Normalization is basically done to remove the differences in the scale of our data provided and make a reference scale which can be applied to all the data at once and still preserve the original meaning of the data. The quantile normalization is also done to limit the maximum and minimum or the data we need to use further, and helps in removing outliers to make data cleaner and have less signal to noise ratio. In our PC analysis, we got 4 outliers, 2 in the bottom left and 2 in the bottom right. And PCA is a classic dimension reduction approach, and it was used to construct linear combinations of gene expression. And through this analysis, PCA scatter plot graph was produced. Now, the plot does not show any considerable distinction between the two groups if we consider the outliers, and it was less fruitful than anticipated. If we remove these two outliers that are, as you can see here, the bottom right and the bottom left, if we remove these two outliers, we can see some distinction present in the lean and the obese types. The DEG pathway and GO pathway. Now, the DE sequence analysis is the analysis of count data from RNA sequence for the detection of differentially expressed genes. Enrichment analysis identifies classes of genes or proteins that are overrepresented in a large set of genes or proteins and may have an association with the disease phenotypes. Now, in this figure, we can clearly see the activation of immune response, adaptive immune response, and leukocyte migration carries the most of the ratio of genes affecting type 2 diabetes in this sample. And the dots represent the number of genes involved in the pathway. The top three have as depicted is 220 genes. The p value of the top three gene is 1.38585 e raised to the power 27. Now, the volcano plot. In this volcano plot, we can see the down-regulated genes present. The most down-regulated gene being the ANKRD20 A8P. This gene is associated with inflammation of subcutaneous adipose tissue. The DEG analysis shows that activation of immune response is the most weighted pathway which triggers adaptive immune response, leukocyte migration. Now, the second most downregulated gene was SLC27A2 gene, and this gene also acts as a biomarker for overweight development, and these findings are to be further explored in their role of gene expression of type 2 diabetes. Now, the conclusion. The results shown in the GO enrichment analysis show that The most downregulated gene, that is ANKRD20A8P gene, plays a vital role in activation of the immune response. There is also plenty of proof that suggests the same, that the SLC27A2 is a biomarker for ovary development. Apart from these, SPX and TTC36 were also distinguished and are showing distinction in the volcano plot. Now, SPX levels are positively influenced by glucose intolerance in pregnant women with gestational diabetes mellitus or GDM, while they decrease in control in women without GDM. SPX is a novel regulator of lipid metabolism in murin 3T3L1 as well, and human adipocytes as well. Through this, we conclude that the data stands true and a connection between adipocyte tissue and blood glucose level regulation exists. Thank you.